ready? We're live? Okay. So it's my pleasure to introduce the next keynote speaker, Dr. Qin Wang. Qin did her MD in China. She got her PhD at the University of Iowa and did a postdoctoral fellowship at Vanderbilt University. And she's now professor of Department of Cell Developmental and Integrative Biology at the University of Alabama at Birmingham.
Can't hear you, Chris.
Sorry about that. Oh, great. No. Yeah, so I'm introducing Dr. Qin Wang.
Still can't hear.
As our next keynote speaker. Qin got her MD in China and did her PhD at the University of Iowa and a postdoctoral fellowship at Vanderbilt University. She's now professor of in the Department of Cell Developmental and Integrative Biology at the University of Alabama at Birmingham. And she's talking about a subject that's very dear to my heart, the adrenergic link between amyloid and tau. And the part that's dear to me is the link, not necessarily the adrenergic part. So it's a great pleasure to have Dr. Wang with us. So without further ado, here she is.
Thank you, Chris, very much for the introduction. And it's really a true honor and privilege to be here to deliver this talk. And I'm going to share with you some of our recent study on the adrenergic link between A-beta and tau. So we're all familiar that Alzheimer's disease is truly a devastating disease, and today we still don't have very effective treatment or any effective treatment for it. In the past few days, we had a number of very exciting and inspiring talks about Alzheimer's disease beyond A-beta and tau. And here I'm going to actually get back to A-beta and tau, and which are the two very essential components of the AD pathology. So based on the amyloid hypothesis, a very simple model is that the AD is initiated with A-beta accumulation, which serves as a trigger, which can induce a cascade of events that eventually cause neuronal degeneration. So one of those events is tau hyperphosphorylation, which is also essential for neurodegeneration. So you can consider tau as a bullet, and then eventually hit to the neuron and cause degeneration. So however, over the past many years of effort, we still have, there's still much unknowns in each of these steps. So we still don't have a clear molecular mechanism that can explain how A-beta actually induced tau hyperphosphorylation and pathology. And also, there are little is known that why certain types of neurons are more vulnerable to degeneration, to the tau induced by the tau toxicity. And also, in the past many years, we all aware that there are many clinical trials that directly target to A-beta actually have failed. So after this talk, I hope I can convince you that norepinephrine, we think, is a key missing piece of this Alzheimer's puzzle, which can help us understand many of these challenges. So a little bit background about norepinephrine. So norepinephrine is a neurotransmitter that is released mainly by the LC noradrenergic neurons. So you can see LC is this very small nucleus located in the brainstem and can send out widely spread projections, long projections throughout the whole brain. And norepinephrine acts on adrenergic receptors to modulate cognitive activities in the cortex, including arousal, attention, and memory. And also, norepinephrine can reduce chronic inflammation. And another thing is very quite striking is that these neurons are particularly vulnerable and in Alzheimer's disease, and they degenerate very early in the disease stage. So, however, now we know that noradrenergic dysfunction in Alzheimer's disease is actually much more complicated than a loss of norepinephrine input, although there is this massive degeneration. Because there are evidences support that the LC degeneration, although they lead to reduction in tissue levels of norepinephrine, when you look at the extracellular level of norepinephrine, which are more relevant to the physiological function of NE, they are actually sustained or even elevated, especially at the early stages of the disease. This is mainly because of the massive compensatory changes after the noradrenergic degeneration. And also, when we look at the early stage of the disease, right, we found that noradrenergic dysfunction actually occurs long prior to LC degeneration. And there are evidence suggests that there is increased tonic discharge of noradrenergic neurons in early stage of AD. So all this have left us to think about whether these noradrenergic neurons, right, they are just simply target or victim of the A beta and tau toxicity, or actually they can also act as a better in this whole process to promote AD pathogenesis. So to address that, we focus on the adrenergic receptors, which are the mediators of norepinephrine function. So there are 3 subfamilies of ARs, the alpha-1, alpha-2, and beta ARs, and each of which consists of 3 subtypes. And these receptors, they have distinct yet overlapping expression patterns in the brain, and they have different affinities to norepinephrine, actually. So you can think of that in case of changes in noradrenergic neuron discharge, or the overall the activity of these neurons, and will actually lead to a imbalance of the reactions from these various adrenergic receptors. So our focus has been on the alpha-2A subtype of AR. This subtype has the strongest affinity to NE and responds to the tonic norepinephrine, and it's broadly expressed in the brain and at both pre and post-synaptic compartments. And another thing is highly enriched in LC noradrenergic neurons. So just keep that in mind. I'll get back to that. So the first thing we want to ask that whether there is any association between the alterations of alpha-2A receptor and AD. So we look at the receptor activity in postmortem tissues. So you can see from this figure that compared to the control subject, in AD patients, you can see there is a significant enhancement of the receptor activity. And then we reason that since there are actually a number of alpha-2 receptor ligands has been used in clinic, we thought to look at whether there is evidence to support the usage of these ligands in Alzheimer's disease. What's the effect of them? So we turn to the NACC database. We simply ask that in patients, right, they've been using the alpha-2A receptor ligand, whether there's any effect on their cognition. So we found that clonidine, which is alpha-2A receptor activator, we found the usage of clonidine actually is associated with a significant reduction of the cognitive function in the demented people, but not in the normal subjects. And furthermore, the detrimental effect of clonidine, or the adverse effect of clonidine, is actually more severe in those patients with more severe cognitive deficits, as shown in panel C. So all this data together suggests that in the AD patients, there is a hyperactivity of alpha-2A receptor, and activation of this receptor actually is detrimental to cognitive function. And then we tried to study the animal models, and we were able to recapitulate the enhanced alpha-2 receptor activity in two independent AD models, the APP-PS1 transgenic model as well as a APP-knocking model. You can see here that this leftward shift of this dose response curve indicate a higher activity of the receptor in the transgenic mice at the 7.5 month old of age, and compared to the non-transgenic leader mice. And this change in alpha-2 receptor activity is actually quite specific, because when we look at another GPCR, the adenosine A1 receptor, we don't see this difference in the receptor activity between the transgenic and non-transgenic controls. And also, this change in receptor activity, we found that is age-dependent, because when we turn to the 5-week-old animals, we see that now we lose this difference between the transgenic and non-transgenic mice in alpha-2 receptor activity. So this data suggests that the change of alpha-2 receptor activity is unlikely due to the expression of those transgenes, but rather related to A-beta accumulation in those mice. So then we directly examine the effect of A-beta on alpha-2 receptor activity. So here on the left channel, we directly add the synthetic A-beta 42 oligomers, which are the toxic species, to the system. We can see that it can sufficiently turn the curve to the left, and means it can increase the receptor activity. And then we see the same effect with the TBS extract as prepared from 80 patients' brains. And you can see that, but when the A-beta is depleted from this extract, and then the effect is lost, suggesting the A-beta is essential for this regulation. So this data suggests that the changes in alpha-2 receptor response sensitivity are indeed caused by A-beta in the brain. So all this motivated us to ask the mechanism. So we asked whether A-beta oligomers actually can directly bind to the receptor. And by several different methods, as I shown here, you can see that indeed oligomer A-beta 42 shown on the top left panel, but not the monomer, actually binds to the cell surface A-beta as shown, detected by this flow cytometry assay. And also the cell surface A-beta and alpha-2A receptor indeed co-localize, as shown in the left bottom panel. And then we measured the affinity of this binding of our A-beta to immunoacylated alpha-2A receptor. We see that the affinity is around 30 nanomolar. And again, this binding is rather specific, because when we look at two other GPCR, one is the alpha-2C subtype of the adrenergic receptor, another again is the adenosine A1 receptor, you see the A-beta actually does not have a significant binding to these receptors. So in order to understand the molecular basis of this A-beta binding to the alpha-2A receptor, we performed the in silico modeling study. So our model predicts that A-beta actually binds to the third extracellular loop of the receptor. So we examined this prediction by mutation studies, and then you can see that when we substitute the loop with all alanines, then the A-beta can no longer bind to this mutant receptor, as shown here by the binding assay. And we also know that norepinephrine binds to a different pocket, and this D113A mutation can disrupt norepinephrine binding. However, when we can see from this binding curve that it has no effect on A-beta binding to alpha-2A receptor. So this data also suggests that the A-beta 42 oligomers bind to an allosteric site of alpha-2A receptor, and also this NE binding and A-beta binding are two independent events. So although there are two separate events, but these two events actually can affect each other. So as shown here, that we see with the presence of norepinephrine, we can see that norepinephrine actually can dose-dependently increase the A-beta binding to the receptor. And vice versa is true for A-beta to norepinephrine binding. So this data not just represent a very signature regulation of allosteric ligands to the GPCRs, but also suggests that A-beta actions, actually the sensitivity of A-beta actions can be regulated by an endogenous neurotransmitter. So here, then we further examine the complex formation between A-beta and alpha-2A receptor in vivo in the mouse brain. Here is in the transgenic APP-PS1 mouse brain. We can see a nice complex formation detected by the co-IP assay here. And then, so now we know that A-beta can bind to the alpha-2A receptor and its activities are upregulated in the AD model and in human patients as well. So then the question is whether this receptor is indeed involved in AD pathogenesis. So the very first experiment is rather straightforward. We did is that we just crossed the APP-PS1 model with the alpha-2A conventional knockout mice. And then from this staining, you can see that in the mice with no alpha-2A receptor expression, there is a significant reduction in A-beta accumulation. So it suggests that indeed A-beta, alpha-2A receptor can regulate A-beta generation. So just to make a long story short, I just want to use this model to summarize our mechanistic findings. We know that the decisive factor of A-beta processing is actually its trafficking. So the APP, when it's located in endosome, it can be cleaved first by beta secretase and then by gamma secretase to generate A-beta. However, there is a very important antagonizing mechanism against the A-beta generation is actually this APP retrograde transport from endosome to Golgi. This process is mediated by a protein, a neuronal trafficking receptor called SOLA. And SOLA can bring APP and retromorph to form a complex to bring APP from endosome back to Golgi. So APP cannot be digested or cleaved by beta secretase to generate A-beta. What we found is that when the alpha-2A receptor is activated through the G protein signaling, it can actually cause a disruption of the interaction between APP and SOLA. So as a result, there are more APP get enriched in endosomes and to eventually generate more A-beta. This is actually represents the first mechanism that SOLA-mediated regulation of APP trafficking can be regulated by a neurotransmitter signaling. So I showed you earlier that A-beta can bind to alpha-2A receptor to increase its activity. Now I mentioned that alpha-2A receptor can promote A-beta generation. So this will form a positive feedback loop to enhance the overall A-beta production. And to test this idea, we performed the experiment in primary culture neurons, as shown here. You can see that we put the human A-beta in the cell culture medium and measured the endogenous mouse A-beta generation. You can see the norepinephrine activation of the alpha-2A receptor can enhance the A-beta production. And then the presence of the human A-beta can enhance the ability of NE to do so. So we further tested whether this is indeed the A-beta binding to the receptor is an essential event to modulate this whole process. We examined the mutant receptor I showed you earlier that A beta cannot bind to. And you see, compared to the wild-type receptor, now in the mutant cells expressing this mutant receptor, now A beta cannot promote itself production anymore. And we further tested this idea in vivo. So we injected human A beta to dorsal hippocampus and then measured the mouse A beta generation in this region. And we see that from this quantitation, you see indeed the human A beta injection can increase the endogenous level of mouse A beta. And if we block the alpha-2 receptor with a blocker called edoxaxane, you can see it can partially reduce the A beta generation. So provide the support to our prediction that this positive cycle actually is very important for A beta to promote the self-renewal. So this is all great data, and then so we studied GPCRs. alpha-2A receptor is a G protein coupled receptor, and we know that allosteroid ligand binding not only increase the receptor activity, but also oftentimes it can change the receptor signaling specificity. So we further asked that whether allosteric binding of A beta oligomer can also affect alpha-2A receptor signaling. So to address that, we performed a kinase array, and we just simply asked whether there's any kinase activation or inactivation only can be observed in cells that co-treated with norepinephrine and A beta. So we're looking at specific signaling can be triggered by A beta and E together. And from our screening, we identified that the co-treatment of NE and A beta can lead to a decrease of GSK3 beta phosphorylation, which indicates activation of this kinase. And you can see here in primary neurons, if you treat the neurons with only A beta or only norepinephrine, you don't see activation of this signaling. But only when both A beta and NE on board, you can see there's a significant reduction of GSK3 beta phosphorylation, which indicates activation of this kinase. So our data suggests that A beta oligomer binding indeed redirect alpha-2A receptor signaling to GSK3 beta activation. And we got quite excited about this because GSK3 beta is a prominent tau kinase, and it's hyperactive in AD brains as well as in AD models, and it can phosphorylate tau at multiple AD relevant sites. And also inhibition of GSK3 beta can reduce tau hyperphosphorylation. For example, in this very elegant study done by Choi and Tanzi and the colleagues, they found that in 3D culture, human neural cell culture model of AD, in the differentiating neurons that express this the familial AD mutations, you can see in the first column, there is a significant enhancement of high level of tau, phosphorylated tau aggregates. However, when they treat these neurons with GSK3 beta inhibitors, you can see that can prevent the tau hyperphosphorylation in this model. So GSK3 beta has been proposed as a critical link connecting A beta and tau. But the question is how A beta activates GSK3 beta. Because in culture neurons, it has been long observed that in order to activate GSK3 beta by A beta, at least or more than 2.5 micromolar of A beta oligomers are required. But this cannot explain how A beta can activate GSK3 beta at the early stage of the disease, because this micromolar range of A beta only occurs when the disease progresses to the pretty late stage. And you can see that through binding to alpha-2A receptor, now we see nanomolar range of A beta can sufficiently activate this critical kinase. And this concentration here we found is actually even less than 1% of the concentration required for GSK3 beta activation in neurons by A beta alone. So through this new mechanism, nanomolar V beta can hijack alpha-2A receptor signaling to activate GSK3 beta. And then we further validated tau hyperphosphorylation downstream of this alpha-2A receptor-mediated GSK3 beta activation, as is shown here. And also, we tested whether this A beta binding to the receptor is indeed required for activation of this signaling. You see here again, we tested cells expressing the mutant alpha-2A receptor that A beta cannot bind. And then you see cells expressing this mutant alpha-2A receptor, now co-treatment with A beta and norepinephrine fails to induce any GSK3 beta activation, tau hyperphosphorylation. So suggests that indeed the A beta binding to alpha-2A receptor is required for this signaling process. And then we go to in vivo again, then we injected human A beta to the dorsal hippocampus and look at the signaling change. And you see that human A beta injection can indeed induce the GSK3 beta activation and tau hyperphosphorylation as highlighted by the red circle. And when we treat the mice with alpha-2A receptor inhibitor to block the endogenous norepinephrine alpha-2A receptor activation, you see that we can successfully block activation of this signaling. So it suggests that A beta indeed hijacks norepinephrine alpha-2A receptor signaling to activate the GSK3 beta tau cascade in vivo. So, okay. So just to summarize what I showed you of this molecular pathway, we actually identified a direct role of norepinephrine signaling in connecting A beta and tau. And you can see under the normal condition, norepinephrine bind to the alpha-2A receptor to induce the normal noradrenergic signaling. However, when A beta is on board, it can bind to alpha-2A receptor at the allosteric site and work together with NE to redirect NE signaling to this toxic or pathogenic GSK3 beta tau cascade. And I want to point out that this A beta, the concentration of A beta, we only require nanomolar range. And this is actually can help us understand the failure of multiple trials targeting A beta removal, because even you have the advanced stage, you already have micromolar range of A beta present. Now you target A beta generation or remove A beta from the system, but if you have the residual A beta left at nanomolar range, you can still activate this pathogenic pathway through binding to alpha-2A receptor through this mechanism we identified here. But on the other hand, if we can inhibit alpha-2A receptor by idazoxan, which is an antagonist of the receptor, we were able to actually block the GSK3 beta activation and tau hyperphosphorylation. So now I show you that this endogenous alpha-2A signaling through NE and A beta can increase A beta generation and promote the tau hyperphosphorylation. We want to test this consequence in the AD model related to the AD pathology, and also to test the therapeutic potential of this pathway. So we treated the mice, the two different AD models, APP knocking and APP PS1 transgenic mice, at a rather late stage at 8 months old age, and then we treated, at this age is already after the A beta pathology is very profound, because we thought this is more closely to the clinical situation. And we treat this mice for 8 weeks with idazoxan or saline, and then allowed a 1-week washout period before we test the change in behavior and pathology. I also want to point out that Atazoxan is actually a clinically tested alpha-2 receptor antagonist. So you can see that in the mice, just show you an example here with the APP-PS1 mice, atazoxan treatment can significantly reduce A-beta accumulation. And also it can reverse the hyperactive GSK3 beta activity in the AD model. And importantly, it can reduce tau hyperphosphorylation. So shown here, you can see the red color indicates the tau, phosphorylated tau, surrounding the green, which indicates A-beta. This is all immunostaining. You can see in the atazoxan-treated animals, the same level of A-beta accumulation actually can cause a reduced level of tau hyperphosphorylation. So the tau hyperphosphorylation reduction in this will indicate a preservation of the brain function. So we further tested the behavior, the cognitive behavior in this mice. And in the APP-PS1 mice, we performed this water maze test. And you can see this, at this age, this mice actually, compared to their non-transgenic, their cognitive deficit is very obvious. However, in the mice treated with atazoxan, you can see at the end of the training, they can behave almost comparable to their non-transgenic leadermates. And same here for the probe trial. And I want to also mention that because we actually integrated a 1-week washout period for the drug, so we think what we see here is not due to acute drug effect, but rather we interpret that as a result of the improvement of the pathology. So we also look at the APP knock-in mice. In this mice, we tested the passive avoidance, and you can see again atazoxan treatment can rescue their cognitive deficits. So finally, we look at the degeneration of LC neurons, because as I mentioned that LC neurons, they degenerate early in human, and in this APP-PS1 model, we were able to see that a similar fiber degeneration as well. Here we are looking at the CE3 of hippocampus. We can see that the APP-PS1 mice has significantly lower level of noradrenergic fibers in this region. However, when the mice were treated with atazoxan, you can see that the fiber degeneration was prevented. So this, I think, our data also help us understand, why these LC neurons are so sensitive to A-beta and tau toxicity. As I mentioned earlier, that these neurons there are a high level of alpha-2A receptor expressed in these neurons. So now I show you that if we block alpha-2A receptor, we were able to reduce A-beta deposition, attenuate tau hyperphosphorylation, and improve cognition, as well as prevent LC neuron degeneration. So one thing is that the alpha-2A blockers, there are several of them already tested in clinic. So redirect these blockers for AD treatment could be an immediate treatment for this disease. So finally, I want to thank people who make these studies impossible. So this study being mainly performed by Fang, Mary, and Inja, and Shalini in my lab. And also we have just wonderful collaborators at UAB and outside UAB who are very important for our study. And also important thing is the funding, of course. And thank you for your attention. I will address any questions.
Fantastic work. One day you will have a very big audience. What are your future plans for the clinical trial with the inhibitors?
Yes, obviously we're very excited about this arbitrary receptor as a novel target, and so we are attacking that by several ways. So one is to push to clinical trials. So our plan is to apply the NIH grants for the clinical trials. And also, we are collaborating with industry to try to get this to trial as well. The nice thing about idazoxan is that it's already been tested in human, and the safety is not it's not an issue. So the bioavailability, safety is all already tested.
Is it an expensive drug?
Sorry?
Is it an expensive drug?
Is not in the market yet. So it's originally tested for like major depression, but it got dropped off because of less of efficacy there. But I think we can actually pick that up for AD. That's going to be promising.
Great talk. I had a quick question because it looks like, and sorry I showed up late, but it looked like there's this interaction between sort of A beta and norepinephrine at that receptor. So I was curious, right, because clinically, a lot of our Alzheimer's patients have mood symptoms or behavioral symptoms, and sometimes we're treating them with antidepressants that include sort of the norepinephrine reuptake inhibitors. Would we expect, per this mechanism that you laid out here, that that might actually worsen their clinical symptoms?
Yeah, so that's totally possible. So I think overall for the, in addition to depression, I think that's a separate topic we can talk about, whether norepinephrine or in depression, whether there is indeed a loss of norepinephrine or lack of norepinephrine or not. But in AD, certainly I think there's definitely, instead of loss of norepinephrine in the brain, is not the case. So especially if you look at the early disease stage, there's rather maybe an increase of especially extracellular norepinephrine. So in the case of people taking the NAC inhibitor, for example, right, or to increase in norepinephrine levels, I guess that can be complicated because it depends on the level, the actual NE level, right? So in the early disease stage, there is an increased tonic LC discharge. So there is an increased tonic level of norepinephrine. And that will definitely goes to alpha-2A receptor because alpha-2A receptor is the one majorly responds to the tonic level of NE. But so in that case, you increase norepinephrine level definitely is not a good thing. But if you can increase norepinephrine level to a certain higher level, that enough to boost, engage the other adrenergic receptors, for example, the beta receptors, you could see some beneficial effect. So that actually quite case dependent, I would say.
So thank you, great talk. My question is, you obviously know that there's a bigger dislink between A beta and tau pathology in mice, as well as possibly this human that has a ApoE Christchurch mutation. So do you have any direct evidence that the alpha-2A AR is able to link to that level of pathology or to a more rampant level of neurodegeneration?
That's a great question. There's definitely worth to look further in details. So the currently, especially in the AD case, right, you could have the distribution of alpha-2A change, or you can have the activity of the receptor change as well. And currently, I don't think we have enough evidence to suggest that. But one thing is for sure is definitely in LC neurons, there are high level of alpha-2A receptor, and we see that degenerate early. And the tau pathology show up first, actually, in LC neurons. But for other regions, that's definitely, I think, worth to look, whether there is a correlation of alpha-2A receptor expression and the tau pathology as well. But we currently don't really have data on that yet.
Yeah, so just to follow up to that, do you think there's a block in the the progression of neurodegeneration from LC to other regions in the brain in mice? I mean, I wasn't even aware that there was neurodegeneration in LC in these models.
Right. So there's indeed a degeneration in the mouse model, not just this APPPS1 mice, and we recently looked at the knocking mice as well. So your first part is, so your question, I didn't quite get it. The first part of your question?
Is there a more profound block in the mouse models, and maybe this individual with the Christchurch mutation, between progression of tauopathy, fibrillar tauopathy, and neurodegeneration from LC to other regions of the brain, to hippocampus and then cortical regions?
So I think that definitely, because so there's evidence if you inject the tau, the pathogenic tau, into the LC, and then can spread out through the LC neurons to the brain.
Okay, I see.
Right. So in animal models.
So it's not just the transgenes.
So no, not the
Okay, got
transgenes.
it.
Sorry, yeah. So yeah, right. So it's not just the transgenes of that, and the LC. So I think the LC integrity would be very important. So if you have the impaired LC, and then there's a massive compensatory change, so you end up have a massive dendritic sprouting, and then downregulation of NAT and upregulation of TH, the tyrosine hydroxylase, to make more norepinephrine. So those all lead to you have actually a hypertonic discharge or hypertonic activity of the system, but you have impaired phasic activity of the system. So that will cause the cognitive function is impaired quite severely. I think another thing is, although we don't have direct evidence, but at least in the APPPS1 mice is probably the one with a very, so when we compare the APPPS1 and APP knocking mice, the LC degeneration occurs early in the APPPS1 mice, and you also observe very early and more profound cognitive deficit there as well. But if we can prevent that, we certainly much improve their cognition, yeah.
Got it. Thank you. Thank you so much for your talk.
Thank you.